shaded portion. Okay. So when the next round of replication is happening, the G is normally present there. So the G again it has mispaired with another thiamine. But the thiamine which is present here, okay, the thiamine which is present here, it will normally pair with a adenine. So look, compare the sequence of this replicated DNA with the parental DNA. At the third position, when you are actually expecting a G and C sequence or G and C pair, here you will be getting a and T pair. So this mutation is called as spontaneous mutation. Okay. So this part, is it clear to all, or anyone is having any questions? So then coming to induced mutation. So induced mutation means when there is some external agent or external factor that is present in the environment, which is responsible for increasing the rate of mutation beyond the spontaneous rate. So spontaneous mutation will occur naturally. As I told you, the rate is actually very or extremely low or extremely slow rate of mutation. It occurs very rarely, like maybe 1 upon 1 lakh sequence. Like if 1 lakh times the DNA is being replicated, there might be possibility that one time the error might occur. But induced mutation is what? Now, when due to some factor or some agent, the rate of mutation increases beyond the rate of spontaneous mutation. So let's see, maybe it is like around 1 upon 100 or 1000, something like this. Means among the 1000 or 100 nucleotide sequence replicated during the DNA synthesis, one of them will be having a mutated or error base. Okay, so that is what is called induced mutation. So what are there are actually two group of mutagens which induce the mutation. One is called as the radiation mutagen, the other one is called as the chemical mutagen. Okay, so radiation mutagen, mostly our UV rays play an important role in inducing a mutation. So as what exactly or in what mechanism it actually induces the mutation? Now, primarily it will be forming a bond between two pyrimidine residues, okay, which we call as pyrimidine dimer. So if there are adjacent pyrimidines like C or TT that is present in a DNA sequence, or even C and T. Okay. So if a DNA sequence has continuous pyrimidine bases, then and if that sequence is actually exposed to UV radiation, the energy that is acquired by or that is actually given by the UV radiation pushes the DNA bases to form a bond between the two pyrimidine bases in the DNA sequence, forming the pyrimidine dimer. So it could be CC dimer, TT dimer, or CT dimer also. So this actually leads to a distortion in the normal double helical structure of the DNA, leading to mutation. Similarly, there are chemical mutagens also. So there are actually classified into various classes. So one such class of chemical mutagen is called as a base analog. Okay. So base analog means the DNA, the chemical bases or the chemical agents or mutagens, they will be having similar chemical structure like that of a normal DNA base. So we have broadly two different type of base analogs are used. One is called 5-bromouracil and other one is called 2-aminopurine. So these two are the common base analogs which induce mutations. So 5-bromouracil, actually this is a base analog of thiamine. Okay. So where we are expecting in a DNA that the DNA adenine should pair with a thiamine, it may mispair with a 5-bromouracil, because if you look into the structure of 5-bromouracil, it is actually almost identical to thiamine, except at this position it is having a bromine, where the thiamine will be having a CH3 or methyl group. That is the only structural difference between a thiamine and a 5-bromouracil. So when the DNA replication is taking place, sometimes by mistake, instead of pairing with a thiamine, the adenine can mispair with a 5-bromouracil. Now, similar to adenine, as the thiamine exists in two different forms, like the normal and the rare form, the 5-bromouracil can also exist in two different forms, the normal and the rare form. So, accordingly, if during the replication event it will change the form, it may lead to a from an AT to GC transition mutation. Similarly, the 2-aminopurine, it is an analog of adenine. So where a thiamine should normally pair with adenine, if 2-aminopurine is available in the environment, it may wrongly pair with a 2-aminopurine instead of adenine. Okay. So again also here also what you will get from AT to GC transition mutation and vice versa. So just let me explain this part to you with one example, only 5-bromouracil. 2-aminopurine will be similar. So here, see, this is a normal DNA sequence. So what is the base pair here? The T has correctly paired with adenine. So now if you are adding 5-bromouracil in the environment and the replication is occurring, so both the strands will be separated during the DNA replication. So here the T has normally paired with an adenine, but the A, instead of pairing with the thiamine, it has been wrongly paired with a 5-bromouracil. Okay. So now the 5-bromouracil, like a normal thiamine, can change its shift like from rare to from like common to rare form, or that is from keto to enol form, like that. So if it changes the form, so 5-bromouracil, then instead of acting as thiamine, it will act as a cytosine. So cytosine, if it is acting as a cytosine, it will no longer pair with adenine, rather it will be pairing with a guanine. So here you are observing that there is a bond between 5-bromouracil, which is similar or equivalent to a cytosine, that has paired with a guanine. But this guanine is in normal form. So when the next round of replication will occur, the guanine will have a tendency to pair with that of a cytosine only, normal cytosine. So you will have the CG instead of T and A. So where originally the sequence was T and A, or A and T, you can, in whatever order you can read, now it has been replaced by G and C. So there is an AT to GC transition mutation has occurred due to this base analog. Okay. Similarly, here if you observe, originally the sequence was C and G. So if you are adding 5-bromouracil in the environment, the 5-bromouracil can be similar act to cytosine. So here the guanine may pair
Madam,
with
5-bromouracil is a chemical. We are adding it.
yes,
Or
that
what?
is a mutagen. Okay, that is a purine like the adenine and guanine. Sorry, like the cytosine. It is a I forgot. That like
Uracil.
the thiamine. No, thiamine. Like a thiamine, it is a chemical only. Okay, so it is a modified form of uracil. You can also say 5-bromouracil. Okay, so it is actually a pyrimidine, a chemical compound that is pyrimidine. Okay. So if it is available in the environment, it's not exactly that like experimentally you can add, or if by chance also you get exposed to this kind of chemicals that is present in the environment, then also this problem will occur. So here the 5-bromouracil will pair with a guanine. Like if it will change the form, then it, instead of acting as a thiamine, it may act as a cytosine. Sorry, instead of acting as a cytosine, it may act as a thiamine. So thiamine, if it is acting as a thiamine, it will pair with a adenine. Okay. So then again, if the form changes, then the adenine, it will behave as a thiamine, and instead of pairing with the guanine, it may mispair with that of a thiamine. Sorry, adenine. Okay. So now in the next round of replication, the adenine will normally pair with that of a thiamine. So ultimately, where you started with a sequence that had C and G, you are getting a copy of DNA that has T instead of C and A instead of G. So CG or GC to AT transition mutation has occurred. See, in order to like remember this thing, you just keep in mind this particular slide I showed you. The rare and the like the Watson and the non-Watson Crick base pairing. Okay. So this slide everybody understood or not, this particular slide, because the entire explanation is based on this slide only. Normally we know that AT and GC bond should be there in the DNA. Okay. But sometimes, if these DNA bases, due to tautomeric effect, change their structure or change their bond, or simply they undergo bond rearrangement, then they can form non-Watson Crick pairing, like the guanine instead of cytosine can pair with a thiamine. Similarly, okay, thiamine I am doing like this. So this means this is the uncommon form or the rare form of the thiamine. Okay. Normally guanine will pair with a cytosine, but sometimes if the thiamine is in a uncommon form, the guanine can also mispair with that of a thiamine. Similarly, in normal condition, the adenine pairs with a thiamine, but if the guanine can exist in a rare form, the thiamine can also pair with a guanine. So this kind of non-Watson Crick pairing is responsible for the mutations. Okay. So this part everyone understood or not? See, this might sound a little bit confusing actually, but if you continuously read for means if you revise it for 2 to 3 times, then it will be easier for you to understand. A simple thing, you have to remember
Yes,
that
ma'am.
A should be pairing with thiamine, C should be pairing with guanine. But if any of these four bases will be changing the shape, means will be undergoing the bond rearrangement due to the tautomeric effect. So tautomeric effect means where the double bond gets rearranged. Okay. So in that condition, their potential to bond with the respective complementary base changes, and accordingly they may pair with the wrong purine and pyrimidine respectively. But always the pairing will be between purine and pyrimidine. Okay, like A is the purine that pairs with the pyrimidine thiamine, but sometimes the A might mispair with the cytosine. The and accordingly the rest of the arrangement will be occurring like that only. Okay, so just you remember, if any of the four bases is changing the form or is changing the means is changing due to the tautomeric shift, then instead of pairing with the normal base, they will be pairing with the rare form of the base, leading to a non-Watson Crick base pairing. Now if in like whatever non-Watson base pairing happens during the next round of replication, it may bounce back, means it may convert from normal to rare to common form. Again, in the common form, it will have a tendency to pair with the normal base. So there only the mutation will occur. Okay. So like we saw here, I'm just explaining it once again. So here it is actually clearly written. Also, everything is clearly explained here as well. So normally you have a AT pair. Okay, so there is no means confusion here. The A has paired with a T and Watson-Crick base pairing. Now, due to presence of the 5-bromouracil, so 5-bromouracil is what? This is a structural analog to your thiamine. Okay. So, like it will appear similar like a thiamine. So, the adenine may wrongly pair with a 5-bromouracil instead of a thiamine, because thiamine and 5-bromouracil will look similar, okay, or they will look alike. So, instead of pairing with a thiamine, the 5-bromouracil has mispaired with a, the adenine has mispaired with a 5-bromouracil. Now, this DNA has no problem, so it will have a normal replication as like it will have a normal replication procedure. But what will happen to this strand? So when during the replication these two strands will separate, The adenine has no pair, no problem. It has a common form and it will normally pair with that of the thymine. So this DNA strand also has no problem. But now the strand which is having the 5-bromouracil. So if like the normal DNA basis or like the purine and pyrimidine basis, the 5-bromouracil may change the form. So where it normal form it looks like a thiamine, in the rare form it will behave like a cytosine, the other pyrimidine. Okay. So if it changes the form from normal to rare form, now it is behaving like cytosine. So in cytosine form, to which it will bind? It will bind to guanine only. Okay. So now the 5-bromouracil in the rare form has bound to the guanine. But this guanine has no problem because it is in the normal form. So it will have a tendency to bind with what? It will have a tendency to bind with the normal cytosine. Okay. So now look at after 3 or 4 rounds of replication, look at the change in the sequence where you had started with thiamine and adenine, that has been replaced by the base cytosine and guanine. So here it is an AT to GC or TA to CG transition mutation has taken place. And now is it clear to all? The same thing can be seen in the second case also where there is a GC to AT transition mutation. So the base analog can induce both type of transition mutation, that is AT to GC as well as GC to AT. Similarly, there are other mutagens also. They are called as the base modifying agents. So they will also induce mutation. So some of the common base modifying agents are here. Okay. So one of the common base modifier is your nitrous acid or HNO2. Okay. So what it can do to your normal bases? It can convert the guanine to a compound called a xanthine. Okay. So guanine is normally pairing with what? Guanine is
Ma'am,
normally pairing with
may
the
I ask a question?
yes.
Ma'am, 5-bromouracil can bind both guanine and adenine.
Yes, but in two different forms. Okay. 5-bromouracil is actually a structural analog of thiamine. That is what I told you. You just keep this one in mind. This slide, if you keep in mind, it will be easier for you to understand that part. Okay. So I request all of you to be a little attentive and focus on this particular slide. Okay. See here, what is this base? This is thiamine, right? So thiamine is pairing with what? Adenine. As you know, the normal Watson-Crick base pairing this is. But the same thiamine is also pairing with guanine or not. The second
Yes.
case, if you observe, okay. So similarly, the 5-bromouracil will act in a similar way. So normal in
Okay.
the normal form, it will pair with what? Adenine, because it is same or it is equivalent to what? Thiamine. It has same structure like that of thiamine. The only difference is what? In the thiamine, here you are having a CH3, that is a methyl group, but in 5-bromouracil you will have a bromine instead of this methyl group. So structurally they are alike. Okay. So like in the normal pairing, it will pair with adenine, but if like as the thiamine, here the thiamine is normal, the guanine has undergone the guanine has undergone the tautomeric shift, but similarly the thiamine, if it will undergo tautomerism, instead of pairing with the adenine, the thiamine can pair with guanine. The same thing applies to 5-bromouracil as well. Okay. So when
Ma'am,
it is
the
in the normal form,
5-bromouracil also show the tautomeric effect.
yes, all the purine and pyrimidine, since they are having double bonds, so they will have their own rearrangement, which we are calling as tautomeric effect. Okay. So it will also have the tautomeric effect. Because of that only there is this kind of changes are happening. So I think here you can observe. Okay, this slide, is it now visible? The same thing is being explained here. Okay. See, this is the rare form of the thiamine. So thiamine normally is in the previous slide what you saw, it was pairing with what? Adenine. But in this condition where it is pairing with normal guanine because it has undergone tautomerism. The same thing applies to 5-bromouracil as well. The similar concept is applicable to 2-aminopurine, where the 2-aminopurine is actually a structural analog of adenine. Like the 5-bromouracil is the structural analog of your thiamine, the 2-aminopurine is the structural analog of adenine. Okay. So similarly, it will have a T pairing, like the 2-aminopurine can normally pair with a thiamine, and again in the rare form it can also mispair with a cytosine, leading AT to GC as well as GC to AT transition mutations. Anyone else has any other questions? Actually, for the first time it will appear very confusing, but once you memorize this part, like the normal pairing and the non-Watson Crick base pairing, it will get easier to you. Okay. You can simply write down it 3 to 4 times for practice purpose, then you can easily memorize it. Nothing too much complication is there. It just you need to remember. Okay. You need to remember and correlate. That's it. Then coming to the next mutagen, that is your nitrous acid. Okay, so nitrous acid, it actually acts on the guanine. So guanine, it converts the guanine into a product what is called a xanthine. Okay. So like the guanine pairs with a cytosine, the xanthine can also pair with the cytosine. So there is actually no mutagenic effect of nitrous acid on guanine. Okay. But the nitrous acid can convert cytosine into a product that is your uracil. So this is nothing but it is also called as a deaminating agent, because what is the difference that you will observe between cytosine and uracil? This amino group from the cytosine has been removed and it has been replaced by C double bond O. So that is the only difference between cytosine and uracil. So due to the deamination effect, the cytosine has been converted into uracil, where normally the cytosine pairs with what? Guanine. But uracil will behave as thiamine and will mispair with what? It will mispair with a adenine. Okay. So again, the similar transition mutations will occur, like we saw in the previous case. Okay. So during the replication, if instead of cytosine, the uracil is present in the DNA sequence, then instead of binding to guanine, it will bind with the adenine, and ultimately the adenine will have a tendency to normally bind with a thiamine. So when you will start the sequence with C and G, ultimately it will be replaced by T and A. So GC to TA transition mutation will occur. Similarly, the nitrous acid can act on adenine also, where it will convert adenine into a product called hypoxanthine. So where normally adenine pairs with that of your thiamine, the hypoxanthine will be pairing with that of cytosine. Now, again in the next round of replication, the cytosine will have a tendency to normally pair with what? Guanine. So the earlier sequence which you started with A and T will be ultimately replaced by G and C. So AT to GC transition mutation will occur. So next there are few other base modification agent or base modifying agents that will also act as mutagens. One is called hydroxylamine. Okay. So this specifically acts only on the base cytosine. Okay. So where, and it will be converting the cytosine into a product called as hydroxylaminocytosine. Okay. So where the cytosine normally pairs with a guanine, this hydroxylaminocytosine will have a tendency to pair with adenine. So again, like the previous case, the adenine will have a tendency to pair with a thiamine. So ultimately, the sequence which you started at C and G, okay. So the C by the action of hydroxylamine will be converted into hydroxylamine cytosine. So the hydroxylamine cytosine will mispair with that of a adenine, and in the next round of replication, this normal adenine will pair with a thiamine. So ultimately, your C and G sequence is replaced by T and A sequence. Okay. So CG to TA transition mutation. But here one thing you keep in mind, like rest of the mutagens which can induce both the transition mutation, like AT to GC and GC to AT, but hydroxylamine is specifically will induce only GC to TA transition mutation. There is no AT to GC because it will only act upon the cytosine base. Okay. Similarly, there are other base modifying agents like your MMS, methyl methane sulfonate, or EMS. So EMS is a very commonly used mutagen for many kind of experimental procedures. Okay, ethyl methane sulfonate. Okay. So they all actually modify the existing DNA bases, changing their bonding capacity. So where the normal guanine pairs with that of a cytosine, due to the action of EMS or MMS, that is methyl methane sulfonate or ethyl methane sulfonate, there is a modification at this location. Okay, so OCH3 is added due to the activity of methyl methane sulfonate, and this is now converted into O6 methyl guanine. So this has a tendency to mispair with thiamine instead of your cytosine. So same thing will happen if this is the guanine.
Ma'am, these are the cytosolic mutations or they are from outside?
No, no, they are actually mutagens, means they are not present in the cell. They are present in the environment. Okay, so if you by any chance you get exposed to these mutagens, then only these kind of problems will occur. Okay, so they are not present, because if they will present, then the cell can normally regulate. The cell can never function normally. So they will create a lot of problems. Okay, so they are actually external agents, mutagens. Okay, they are not present in the cytosol. So similar to this, there will be GC to AT transition mutation. Similarly, the thiamine can also be modified at the fourth position. Actually, at the fourth carbon position, the thiamine gets modified due to the action of MMS or EMS, and instead of pairing with adenine, the thiamine mispairs with that of guanine, leading and AT to GC transition mutations. So these base modifying agents, these are actually called as alkylating agents. Okay, alkyl, they are actually donating an alkyl group to the DNA base, like ethyl or methyl group. That is why they are called as alkylating agents. So they can also induce both AT to GC transition mutations. Next, coming to another group of mutagen, which is called as intercalating agent. Okay. So you can write down name of few intercalating agents. One is called ETBr, ethidium bromide, which is a commonly used chemical for visualization of DNA. Okay. And you can write down another name, acridine orange. DINE orange. This is a dye actually. So ETBr is also a dye. DINE acridine orange. Okay. So these are actually called as the intercalator or intercalating agents. So they actually induce insertion and deletions leading to frame shift mutations. Just so as you can see here, these intercalator, these chemicals actually they have the same size like that of one nucleotide of the DNA. So what they can do when the DNA replication is happening, they can either insert themselves in the DNA strand during the replication or even in the means resting DNA also. So here if you observe this particular strand, just let me zoom it a little bit. So here, this was actually the original sequence of the DNA, ATCAG So what this intercalator has done, as it was present in the environment, it has randomly incorporated itself or it has inserted itself into the base between G and T. Okay. So now all the other bases of DNA, they have their complementary bases to pair with, but this intercalator, it does not have a complementary base. So randomly any base will be in means placed or will be paired against this DNA intercalator. So what is the new base that is added here? That is G. Randomly, it could be ATCG anything. So let's assume here the base that is added complementary to intercalator is G or guanine. So now when this strand will undergo replication, so both of these strands will separate. So when this strand will undergo replication, the G will have a complementary pairing that is C. So earlier if you look at the sequence, there was no additional CG pair in the DNA sequence, in the original DNA. But now a CG pair has been inserted. So if you look into the reading frame here, definitely the reading frame will be affected. Because if you look into the codon, like earlier it was ATC, AGT, TAC like this. But now if you look, the reading frame has changed. So what is the new reading frame? ATC, AGC, but earlier it was what? AGT. So following this point of mutation, the entire reading frame will be disrupted. So this is actually mutation due to intercalator. This is a kind of insertion mutation. Similarly, it could lead to deletion mutation also. Okay, here as you can observe, so here against the T, where there should be against the T, what should be the base? Adenine. But instead of A, what you can observe, an intercalator has been inserted. So when these two strands will separate and will undergo replication, this intercalator will be again removed because it has no role in the DNA sequence. So randomly it has been inserted, now it will be removed. So in the original sequence, what you have? You have a T and A base pair there. But in the new sequence, this DNA, as it has been removed, the intercalator has been removed, this pairing will be lost. So this part will be completely lost. Okay. So now you have a change in the reading frame as well due to a deletion. In the earlier case, it was due to insertion. Now it is due to deletion. So these DNA intercalator can induce frame shift mutations. Is it understandable to all? Like everyone understood this part? So this will lead to,
Yes, ma'am.
because of this insertion and deletion, it will lead to a frame shift mutation. Okay. So DNA intercalators are very they are too much dangerous as they can induce both insertion and deletion, leading to a frame shift mutation. So if anyone has any question till now, you can ask, or else we will move to the DNA repair process. Okay, so let me summarize this for you. Actually, the mutation could be two types based on at what rate it is happening. If the mutation is happening naturally on its own, then the rate of mutation is very slow. Like it could be 1 lakh, like 1/1 lakh, or 1/10⁵ basis per gene per generation, means within a generation of individual, one gene might have only one base defect among 1 lakh bases that is replicated or that is synthesized in a DNA strand. But if there are some external agent or there is any mutagen which is present in the environment, it can increase the rate of mutation beyond the spontaneous mutation rate. So in that condition, we will call it as induced mutation. So the induced mutation could be 1/1000 or 1/100. That means a gene might have a base defect among each of the 1000 bases that is replicated or synthesized during the DNA replication. So there are different types of mutagens. One could be the radiation mutagen like the UV, which can induce a pyrimidine dimer in the DNA. So that will also disrupt the double helical structure of the DNA, leading to either DNA break or a mutation during the replication process. Similarly, there could be chemical mutagens like your base analogs or base modifying agent or your alkylating agent. So they will somehow modify the existing DNA bases and leading to the change in the form of the DNA bases due to the tautomeric effect, which will have a non-Watson-Crick base pairing leading to mutations. So most of the chemical mutagen can induce both AT to GC as well as GC to AT mutation. There is only one mutagen, that is your hydroxylamine, which has a specificity, that is it will only induce CG to AT transition mutation. It cannot induce AT to GC transition mutation because this specifically acts on modifying the base cytosine only. Okay. And there are another group of mutagen called as the DNA intercalator, which can insert themselves because they have the same size as that of the nucleotide of a DNA base. So, sorry, of a DNA sequence. So they can insert or they can remove themselves instead of a normal DNA base, leading to insertion and deletions that may ultimately lead to frame shift mutation in the gene where the mutation is happening. So as I told you, the spontaneous mutation rate is extremely low. So one of the reason is what? It is because the cell is capable of repairing most of the DNA. Okay. So there are different type of repair mechanisms. So I have tried to include two different types of DNA repair mechanism. One is called the nucleotide excision repair or NER repair mechanism. Okay. So this is very simple as the steps you can see. So this is actually working on which type of defect? Now this is working on a thymine dimer. Okay. So this is actually a UV induced mutation repair. Okay. So as the name suggests, nucleotide excision repair means a nucleotide. So DNA is also made up, actually DNA is called as a polynucleotide because it is made up of repeating unit of nucleotides. So here excision means to cut. So as the name suggests here, what we can expect, now the part of the nucleotide that contains the means mutated region or that contains the problematic region will be cut or will be excised. So there is a set of enzymatic complex called as UVR, UV response or repair gene you can call. So there is a lot of enzymes which together constitute the UVR family. So they are all involved in the nucleotide excision repair. So first what you can observe, the dimer or the mutant region is located or it is actually identified by the UVRAB complex. Okay, so the damaged site or this pyrimidine dimer site can be identified by the UBR AB complex. Now, as the UBR AB complex binds to the mutagen site, the UVA is released and only B remains bound to the location or leading or following the binding of UBRC. Okay. So now B and C are bound to the damaged site on the DNA position. So the C, it has a endonuclease activity. So what it can do, it can create DNA cuts at both the 5' end as well as the 3' end. Now, after the C induces the cut, there is recruitment of UBRD. Okay, so what the UBRD will do? The UBRD will bind and will unwind the region between the cuts, releasing the cut segment. So, as it is the helical structure, you are seeing it in 2D, that is why it is appearing linear, but it is actually in the helical form. So this UBRD, what it will do, it will unwind and it will try to release the cut segment from the existing DNA strand. So now this will create a gap over here. So the gap can be sealed by the DNA polymerase 1. So the DNA, as there is already a complementary, means complementary strand is already present, so in complementary to this, the the new nucleotide bases can be added and the gap can be sealed, and ultimately the end location or the end position will be sealed by the DNA ligase, making the strand complete. So wherever the damaged portion was there, now it has been successfully repaired. So this mechanism is called as nucleotide excision repair. Similarly, we have a mismatch repair mechanism also. So mismatch repair mechanism means, as we saw, that sometimes there could be non-Watson-Crick base pairing. So here there is a mismatch has occurred. Okay. So what is the mismatch? Let's see. The mismatch is,
And this repairing system occur during the replication or anytime?
no, actually it occurs during the replication. See, no, the UV radiation one, actually it may occur anytime, because it's not necessarily that during the replication only the pyrimidine dimers will be formed. But this mismatch repair, actually it is kind of a proofreading activity of the DNA polymerase. So it occurs mostly during the replication process. Okay. So just
Ma'am, if we got 5 mark question of NER, then we have to draw the diagram.
yes, you have to do the diagram. Also, you have to explain a bit in the means. You must mention like why the DNA is being damaged. So you have to give the reason also, due to the UV radiation and what, why the damage is being happening to the DNA, and how the DNA is respond or the cell is responding to that. So you have to explain it a bit also, and for the best results you must do this diagram that will actually clearly illustrate what the mechanism of the repair. Okay. So this is
Okay.
mostly required. The like, even if you write less, but if you can clearly draw this schematic diagram, that will be also sufficient, because it clearly demonstrates the mechanism, how exactly the repair process is happening.
Okay, ma'am.
So in the mismatch repair, let's say there is A and C. Okay. So
म्यूट करना है चरण म्यूट
there is a okay, just let me what I can say. This actually it's not visible to me only. So let me enlarge the diagram a bit. So this is visible to you clearly, right? All of you can visualize. So this is the position where the mismatch has occurred. So let's see, this is A. So A should pair with what? T, but let's say the mismatch is what? A with C. So this strand is having A, this strand is having C. Okay. So now there is a group of enzyme that is called mute. So you can say the mutation repaired enzyme. So there are many different components of the enzyme, like as you can see, H is there, L is there, S is there, like that. Okay. So what this group of enzyme will do, now they will try to find out, or they will try to rectify where the mismatches occur. Okay. So now just we means pay little attention what you are observing. In the same strand, you are having a recognition sequence. So what is that sequence? GATC. So this is highly essential for the repair to take place. And moreover, what you will observe, the adenine is methylated here because there is the presence of a methyl group. Okay. So this actually, okay, so what happens in our DNA, whatever epigenetic modifications are there, then it is actually present in the template strand. So after the DNA is completely synthesized, then only the methylation is added to the new strand. So that is how you can distinguish which is the template DNA and which is the newly synthesized DNA. Since the newly synthesized, since there is no methyl group present in the second strand, this is actually indicating that this is the newly synthesized DNA strand. And whatever strand is having the methyl group, that is actually the template strand or the original or the parental strand. So how you will identify? Because if you are observing a AC pair, so it could be that either of them could be the wrong base. Maybe it is the A is the wrong. So means it should be G instead of A, or the other way could be A is correct. It should be T in place of C. So how you will identify that which one is actually correct? So for that you should know that in whatever sequence or in whatever strand the methylated GATC is there, that is the correct base. So here, as we are assuming that A is present here, so A is the correct base here. Okay. So next step is what? The mute S will go and find or bind to the wrong side or to the mismatch side where it has occurred. Then the mute S bound to the mismatch forms a complex with mute L and mute H to bring the unmethylated GATC close to the mismatch side. So it is forming a loop kind of structure. So now the mute H nicks the unmethylated DNA strand and an exonuclease excises a section of the new DNA strand, including the mismatch. So here this part is actually made into the form of a loop. This part where there is no methylation in the GATC side. Okay, so this is also actually, this is a palindromic sequence. So here it is also GATC. From this side also it is a GATC strand. Okay, so this unmethylated GATC is brought closer to the your mismatch side, and there is an exonuclease enzyme which can cut this part, this GATC part, because this acts as a recognition sequence for the for the exonuclease. Okay, so along with the mispair base, the entire sequence is cut. So as there is a gap formed, the gap can be now filled by the DNA polymerase with the correct base pair. Okay. So that is how a mismatch is repaired during the DNA replication. So first there is a mute S enzyme which goes and binds to the mismatch site. Then along with two other enzymes, that is mute L and mute H, a complex is formed, and this recognition site is brought close to the mismatch site of the DNA. Then an exonuclease comes and cuts the entire wrong sequence, along with the mismatch sequence, it will be cutting the rest of the DNA sequence as well, for creating a gap where the gap can be now correctly filled by the DNA polymerase 1 and ultimately sealed by the DNA ligase to form the complete DNA strand. Okay. Then coming to transposable elements. So these are some of the
All these repair system is present in the human being.
yes, they are present in humans. Okay, so like not only humans, they will be present in most of the eukaryotic cells. Even the prokaryotic cells can also have this kind of repair mechanism. So next comes the transposable elements. So transposable elements are also called as your jumping genes. So they have a tendency to move within the genome. Means they can move by two ways. There are actually two types of transposable elements. One is called the DNA transposon, other one is called the RNA transposon or retrotransposon. So DNA transposon is it moves by a cut and paste mechanism. Like it will be, if it is present in one location of the genome, it can simply cut itself from that location and move to a new location. And the retrotransposon is what? It will move via a RNA intermediate. Means it will move via a copy and paste mechanism. So the DNA sequence or the DNA transposon will make a RNA copy of itself. The RNA will again make a complementary DNA copy, and that cDNA will go and insert itself into any other place in the genome. So in bacteria also, you have a set of transposable elements. Here there are two examples have been cited. Actually, this is altogether a very different topic. So just know the basic idea that transposons or transposable elements are the jumping genes. They are actually present in the genome of any organism. So they have a capacity to move from one location to another location within the genome of an organism. Then coming to the molecular part, actually the expression part of the gene, the organization as well as the gene expression part. So the basic difference between the prokaryotic and the eukaryotic gene is that the prokaryotic genes, they do not have any introns, and the mRNA produced are also polycistronic. Means multiple genes are present or they are under the control of the same promoter in case of prokaryotes. But in eukaryotes, you have a sequence of coding genes interrupted by a sequence of non-coding genes, which are called as exons and introns. And together, this gene is called a split gene. So if you know, that is fine. If you do not know, please note it down. In case of eukaryote, the gene is called as the split gene because this is actually a combination of coding sequences called as exons interrupted by non-coding sequences called as introns. So together this gene is called as a splitting gene. And in case of eukaryote also, the genes are monocistronic, means a single gene is under the control of a single promoter. Unlike in prokaryote, like in E. coli, as the example is cited here for the maltose gene, there is a single promoter that is controlling the synthesis of three different structural genes. Okay. So this is the difference, and moreover, in prokaryote genome, there is no introns are there. So it is only made up of coding sequences or exons. So this is what I was telling you, the eukaryotic gene. This is also called as the split gene. So they are actually made up of alternating sequence of exons and introns.
One
Someone
gene
is
and
asking
one
something.
enzyme hypothesis can apply in the prokaryotic gene.
Yes, that is applicable to all. Okay, so that means, see, polycistronic means, as I already mentioned here, the gene is under the control of a single promoter, but each gene has its own mRNA and each gene is having its own gene product. Okay, so that is the concept of one gene, one enzyme hypothesis. Means every gene will code for its own gene product. So that is applicable to both eukaryotic also, prokaryotic also. So as the eukaryotic gene is a combination of exon and intron, the introns, they do not have any codon, so they need to be removed from the final mRNA product by a process called as splicing, mRNA splicing. So this is the structural organization of a typical eukaryotic gene. It has actually a lot of more informations are there, so I request all of you to go through your material, like what are the component of the 5' UTR, what is a promoter, or in what is enhancer, silencer. These things are written in detail, so you go through that. If you face any problem or difficulty, you can ask me. Okay. So here, as you can see, this is the regulatory sequence. So regulatory sequence means they will actually control the expression of the gene, like whether the gene should be expressed more or it should be expressed less, or whether it should be expressed at all or not. So they are regulated by the regulatory sequence. So the regulatory sequence includes the enhancer, a silencer region, the promoter region. And there is a core promoter region which is conserved in actually most of the most of the members in a species. Then there is 5' on translated region, 5' UTR, and here comes the start codon. Okay, and then it will be ending with the stop codon. So in between you have the introns which will be spliced during the synthesis of the mRNA. Okay. So following the point of stop codon, there is also 3' UTR, which is also regulatory in nature. Now, there is another important component of a gene regulation, that is called your transcription factor. So the transcription factor also, they have significant roles in controlling the expression of the gene. So broadly, they are divided into two categories. One is called the general transcription factor, other one is called the specific transcription factor. So they actually regulate the binding of the RNA polymerase to the DNA. So ultimately, means controlling the expression pattern of the gene. So here is a list of, so you should remember the function of each kind of transcription factors. Okay. So what exactly they do during the transcription process. So here is a list of general transcription factors. So general means they will be present in or they will regulate each and every kind of gene. Then there is specific transcription factor which are actually specific to regulation of certain type of genes. Okay. Then the gene is also regulated by the means the RNAs also play a major role in the regulation of the gene expression. So as you know, the most important part of RNA is your mRNA, which will actually decode whatever is present in the DNA. Okay. So from DNA you will have the mRNA that will carry the codons. Then the mRNA will be along with the ribosomes will move towards the tRNA, where the tRNA is having the anticodons that will read the codon on the mRNA, and the polypeptide synthesis will occur. Okay. So alongside the mRNA, tRNA and rRNA, which actually synthesizes the ribosomes, there is a list of other RNA like your snRNA, snORNA, then scRNA, then incRNA, microRNA, silencing RNA. Together they are called as RNA interference or RNAi. They also play an important role in regulation of the gene expression. Okay. So that we will discuss in later also, how the RNAi plays an important role in regulation of the gene. Then comes the epigenetic modifications. I think someone was asking about epigenetics yesterday. So epigenetics is, okay, we know that there are DNA sequences, that is the base pair sequence A, G, T, C, A, C, like this. Okay. So this is up to this it is genetics, okay, because this is actually representing a genetic code. Now anything that occurs above the genetic level is called as epigenetic. So if these DNA bases they undergo chemical modifications, like you can observe here the cytosine. Okay. The cytosine has been like there has been a addition of a methyl group at the fifth position of the cytosine. So this kind of modification is called as your epigenetic modification. So epigenetic modification is when the DNA bases or the histone proteins that also constitute the DNA get chemically modified. That modification is called as your epigenetic modification. So there could be different type of epigenetic modification, but primarily two types of modifications are studied for gene regulation, means to understand the gene regulation process. One is called methylation and the other one is called acetylation. Okay. So methylation usually has an inhibitory effect on the gene expression, and acetylation is actually a positive marker for the gene expression. Okay. So as you can see here, as a methyl group is added, the cytosine is converted into 5-methyl cytosine, and we can say that the DNA has been methylated. So here, if you are observing a sequence of DNA, most of the cytosines here, if they are modified or they are added with a methyl group, we can call this sequence as a methylated DNA sequence. Okay. Similarly, as the DNA can be as a methyl group can be added to the DNA, particularly to the cytosine base. Similarly, the acetyl groups can be added to the histone protein. Okay, so mostly in the histone, the lysine and the arginine bases they get modified by addition of an acetyl group.
Ma'am, methylation on the DNA can will be cause any mutation?
No, no. See, mutation is only when there is a change in the DNA sequence. So epigenetic modification does not change the sequence of the DNA. It will remain as it is. So epigenetic modifications will not induce any kind of mutation. So since it is above the genetic level, that is why they are called as epigenetic modifications. Okay. So as the DNA is methylated, the histone is acetylated. So there is the histone chains are there where the histone tails are there where an acetyl group is added. So already the DNA, the histone is having positively charged amino acids like your lysine and arginine, etc. So addition of the acetyl group actually means it increases the positive charge on the histone tail. So they try to stay away from each other. So the more the histones will remain away from each other, the more accessible will be the DNA for the RNA polymerase to carry out the transcription process. So, but in case of methylation, it is opposite. The DNA is carrying a negative charge and the methyl group is carrying a positive charge. So it will form a compact structure like this. So as there will be compactness, the DNA is not available for the RNA polymerase or the transcription factors to bind to. So here it is extremely difficult to transcribe a gene. So when a gene needs to be turned off, it will have a methylation, and if a gene needs to remain active or it needs to be expressed, it will have the acetylation. Okay. So these are the markers of the gene expression. Methylation, negative marker, acetylation, positive marker. Then comes the splicing mechanism, alternative splicing. This is actually one of the very crucial things, particularly for eukaryotic cells. See, the number of protein that is present in a cell is actually much more higher than the number of genes that transcribe the protein. So how is it possible? How a single gene can give different type of gene products? So one of the mechanism is because of the alternative splicing. Okay. So alternative splicing means what? Now during the removal of the intron, different type of exons could be joined to give different forms of the protein. Okay. So this is the basic mechanism of alternative splicing. This is called as ubiquitination process. So as you can see here, so the sequence of the intron is essential for the splicing to happen. So like here, the 3' end of the intron should have, or the 3' end of the exon should have a GU base pair or GU sequence. Similarly, this 5' end of this exon should have a AG. Okay, and in between there could be adenine. So there is U1 SNRNP and U2 SNRNP. By utilizing or by utilizing a molecule of ATP, this U2 gets actually loaded on the U1 gets loaded on the GU sequence, and the U2 gets loaded on the adenine sequence. Then comes another set of enzymatic complex, that is U4 U6, that is one complex, plus followed by U5. So together they will form a complex structure which is called as the spliceosome. So this spliceosome is actually inactive. Now, when with another ATP will be utilized, releasing the U1 and U4, then only it will be converted into an active spliceosome. So what are the components of the active spliceosome? The active spliceosome is made up of U2, U5 and U6. Then there will be a kind of twist will occur where there will be formation of a lariat structure. So lariat structure means exactly how I don't know if you know this, jemti amaro jo gai ku jemti ba bachru ku jemti pogha paka hue, it is like that. Man jemti tome bulle ki jo fasota paka hue, the lariat structure is exactly like that. Seithru ta nada diya hai. Okay. So here this OH group here will be acted upon this 5' end, and the entire structure will be removed. So this OH group what is available here will be, it is actually the 3' end, and the 5' end of this exon will now join with the 3' end of this OH, forming only a continuous sequence of the exons, and the entire set of intron will be released along with the spliceosome concept, along with the spliceosome complex. So that is how the introns are removed by the splicing mechanism. So there are actually two group of introns. One is called the group 1 intron. The second one is called the group 2 intron. So the splicing mechanism is different for both of them. In group 1 intron, as you can see, no lariat is formed here, and here also the sequence could be a little bit different. Like as you observe here, the 3' OH of the guanosine acts as a nucleophile attacking the phosphate at the 5' splicing end. So this is the 5' end, this is the 3' end, and for this particular exon, this is the 5' end, this is the 3' end. Okay. So here as you observe, this 3' end, it is having a OH group. Okay, and what other factor is present here? There is a guanine along with the OH group. Okay, so this guanine along with the OH group will be attacking on this part, and ultimately the 3' OH of the 5' exon becomes the nucleophile, completing the entire reaction. So this entire sequence will be removed. This OH will now act upon this 5' phosphate of this sequence. So the U where it is remaining, here it is, and it will be bonded by the phosphate, and this entire set of intron will be released, leaving only the exons together. Okay, so this is the splicing mechanism of group 1 intron. But the group 2 intron is whatever we saw in the previous slide, that is there is the formation of the lariat structure, there is presence of see, you might be getting confused why it is U, because since it is your mRNA, so instead of your thiamine, here it is U has been mentioned. Okay, uracil has been mentioned. So the only difference between group 1 and group 2 intron is what? Here in the group 2 intron, there is a formation of the lariat structure. No such lariat structure is formed in case of your group 1 intron. Here it is, the entire intron is removed as it is. Now coming to what is the significance of alternative splicing. So as you can observe here, there are two different types of protein has been synthesized. In one of the splicing mechanism, you see the exon 3, the exon 4 has been removed along with the intron. The exon 3 and the exon 5 have combined. So whatever codons are present in exon 3 and exon 5, together they will have a particular type of protein. In another form of mechanism, what has happened, the exon 5 is not included, the exon 3 and the exon 4 has been means clumped together, giving the coding region. So this is highly essential to form different polymorphic form of the protein. So the gene is same. What is this gene? The gene is DSX. So this is actually sex determining gene in case of Drosophila. So, but whether the Drosophila will develop into a male or whether it will develop into a female, it is actually controlled by the alternative splicing of this DSX gene. So, if the splicing happens in such a way that where the exon 3 and exon 5 are together, it will release or it will produce male specific DSX protein and the sex will be specified as a male. But if by the splicing mechanism exon 3 and exon 4 are joined and the exon 5 remains means it is omitted, then the fly will develop into a female as the female specific protein will be produced. So this part also we will see in detail in the next unit actually, where we have this where we have this sex determination mechanism. So that we will see a bit in detail in the next unit only. Then coming to the last part of the unit, that is how to control the expression of the genes. Okay. So as we discussed, there are different types of RNA like your siRNA, miRNA, that is your small interfering RNA or a microRNA, they can control the expression of certain genes. So just have a look at the list of the RNA responsible for like how exactly they will be acting. So together they are called as RNA interference. Okay. So if you compare how the siRNA is different from the miRNA, so siRNA, the base pair is around 21 to 23 base pair, but in miRNA it is around 19 to 25. Now, the complementarity, it is fully complementary to the target messenger RNA. Okay, like if there is a particular gene and that gene is producing the mRNA, the siRNA will have exactly the same complementary DNA sequence that is present in the mRNA. But the miRNA, it will have partially complementary. Okay, so it will have a partial complementary to the target mRNA, particularly it targets the 3' UTR of the target mRNA. So now you can understand why the 5' UTR and the 3' UTR are called as the regulatory sequences. Because if any kind of these RNA interference will bind to the regulatory sequences, then the expression of the gene can be inhibited. Okay. And number of mRNA target, usually it is very specific, only one, because it is completely complementary. But since it is partially complementary, it can target many different mRNA from different genes. Regulatory mechanism, it usually leads to the cleavage of the target mRNA, means simply the mRNA will be broken down, and here there is degradation of the target mRNA, and also the translation can be inhibited. Okay. So let's see how exactly the mechanism works.
Every mRNA and sRNA is present to every mRNA.
No, not necessarily, but it might be present. Okay. It's not necessary that all the mRNA will have their complementary siRNA or miRNA. It depends upon the cell, like how it has developed to regulate its own synthesis. So it might be, means for most it might be present, but it may not be present for all. See, this is not the only regulatory mechanism. This is one of the regulatory mechanism to control the expression of the gene. So it's not necessary that every mRNA in the cell must have a complementary siRNA or miRNA. Okay, so like if we look into the mechanism, how exactly it works. See, this is the large double-stranded RNA molecule, okay, and there is a protein called a dicer. So a large double-strand RNA molecule is diced into the small double-stranded interfering RNA that is around 21 to 28 base pair long. Okay. So the dicer has to dice this double-stranded RNA to form or to synthesize RNA interference. So if the length is like 19 to 25, it will be miRNA. If the length is like 21 to 23, it will be a siRNA. Okay. So the small interfering RNAs and protein, they assemble into a ribonucleo particles, okay, where they are actually assembling near the where the means it is assembling along with the ribosome. Okay. Then the small interfering RNA in a ribonucleotide particle is unwound to produce an RNA-induced silencing complex, which is called as RISC, RISC, RNA-induced silencing complex. So this is actually having your RNA to target the specific mRNA. So this is the mRNA which has a specific complementary sequence to your RNA I or RNA interference. So this small RNA will go and bind to the complementary location on the target mRNA. So if this is an siRNA, it will simply cleave or will break down the RNA here. Okay. Now if this is supposed to be an miRNA, so it will create imperfectly, as it is not completely complementary, so it will be partially complementary, but as it will remain bound to the mRNA, no more translation can occur. And ultimately, it also lead to degradation of the messenger RNA. So if the codons on the messenger RNA cannot be decoded or cannot be translated into the protein, we can say the gene has been suppressed or the gene is not expressed. So is it clear to all, or if anyone has any question you can ask? Then we will move to the last part, that is your operon concept.
Ma'am, gene expression is related to only mRNA or also DNA?
No. Gene expression means the gene is capable of producing a protein. That is what is called a gene expression. When the gene is producing or is transcribed into a mRNA and that mRNA is producing a protein, we call that the gene is expressed. That is what is meaning of gene expression.
So the gene expression can be controlled at the level of DNA?
Yes, DNA, as I showed you, it can be controlled at the epigenetic level also. If it is methylated or acetylated, then also expression can be regulated. Similarly, there are different stages of gene regulation. One could be like in epigenetic modification could be there. Similarly, there could be post-transcriptional modification could be there, where the you saw the alternative splicing. So there also the gene is regulated, which type of protein will be produced, like which exons will be actually coded. So post-transcriptional modification is there, even post-translational modification is there. So there are different levels of gene regulation. Okay. So one such a regulation is this RNAi regulation, where it is actually, you can say at transcriptional level, it is being regulated. Because transcription has occurred, but no translation is being allowed due to destruction of the mRNA.
And this type of gene regulation will lead to gene expression.
Yes, it will control the gene expression. Whether the gene will be expressed or not expressed, that is dependent upon the regulatory mechanism. Like here, as we saw, the RNA interference is suppressing the gene expression. It does not allow the gene to be expressed.
Okay, ma'am.
Okay, any other question?
Madam, what is the difference between translation and transcription?
Transcription is synthesis of mRNA, sorry, DNA to mRNA is transcription. mRNA to amino acid sequence or polypeptide chain is translation. Okay, next coming to the expression of the gene or the regulation of the gene at the operon level. So I hope this is known to all of you, the lac operon and the tryptophan operon. So there are actually two ways by which the operon is regulated. One is called as the positive control mechanism, another is called a negative control mechanism. As you can see in the diagram, the positive control mechanism and activator is there that controls the expression of the gene. In negative control mechanism, it is the repressor that controls the expression of the gene. Okay. So here in the again you can have within either whether it is positive or negative, you can have a inducible and a repressible system. So inducible means when the inducer will means it will deactivate the repressor to allow the expression of the gene. That is called as the inducible operant. Now repressible is when the molecule or the the signal molecule will activate the repressor to switch off the gene. So that is what is called as a repressible system. So as the name indicates, inducible means something is induced, repressible means something is repressed. So if we look into the example of the lac operon, so this is this is the polycistronic because it is a prokaryotic operon. It is present in the E. coli. Now this is the as you can see, this is the activator binding site. These are the regulatory sites. This is the promoter. Then this is the operator binding site, the repressor binding site, which is also called as the operator. Then these are the structural genes that will individually code different enzymes. Okay, so here you can see that one gene, one enzyme hypothesis is there. Okay, so each gene is having its own set of protein. So each gene is coding a single polypeptide. Okay, so there are multiple proteins are produced. That is why this kind of mRNA is called as polycistronic mRNA. So how exactly the lac operon operates? This is the mechanism of lac operon. This must be known to you. Just let me revise it for you. Here, this is an example of the lac operon is an example of uh, inducible negative control mechanism. Okay, so this is an actually inducible operon. So what is the inducer here? Although it is called as the lac operon, the allolactose is the inducer. Okay. So the allolactose is actually synthesized by this beta galactosidase. So now, in absence of lactose, the repressor remains bound to the operator region. So the RNA polymerase cannot move and no mRNA will be synthesized. Now, when lactose is present or due to presence of lactose, the allolactose is synthesized, the allolactose can go and bind to the repressor molecule. Now, due to the binding of the allolactose, the repressor can no longer remain bound to the operator. It will be released from the operator side. So, as the as the repressor is released, now the DNA is accessible to RNA polymerase. So, RNA polymerase will move and mRNA will be synthesized, and we can say that the gene is expressed or the gene is turned on. So, in absence of lactose, the repressor will remain bound to the operator region so that the RNA polymerase cannot move and no mRNA or no expression of the gene will take place. But in presence of lactose, which is converted into the allolactose will go and bind to the repressor. The binding of the repressor to the binding of the allolactose to the repressor will release the repressor. and will allow the RNA polymerase to carry out the transcription. So mRNA will be synthesized. Since here the presence of lactose is increasing the expression of the gene, we call it as a inducible operon. Okay. Now there is a there is a mechanism called as catabolite repression. So you should remember this because this is means this could frequently be asked in your questions. So when both glucose and lactose are present in the medium, so which source should be utilized by the bacteria or which should be utilized by the E. coli? So in that case, glucose will dominate and will try to suppress the expression of the lac operon. This is what is called as catabolite repression of the lac operon of the E. coli. Okay. So when the lactose is when glucose is low, glucose low means lactose will be high. So at that time, the cyclic AMP is also high, lactose is present. So it will operate like the lac operon we saw. So the lactose will remain, allolactose will remain bound to the repressor. The repressor cannot bind to the operator region. So the RNA polymerase will move and mRNA will be synthesized or the lac gene will be completely expressed. But if there is glucose in the medium, then the high glucose actually leads to a low CAMP level. A cyclic AMP level will be low. Even though lactose is present, the glucose will not allow the lac operon to be expressed. So here, so actually why this happens? Now because of low cyclic AMP, this binding of the RNA polymerase to the DNA strand cannot take place because the cyclic AMP plays an important role in binding of the RNA polymerase to this operator region. So if the cyclic AMP is low due to presence of high glucose, even though lactose is present, the gene expression will be suppressed. Okay, so that the organism is forced to utilize glucose first before utilizing the lactose. So this kind of suppression of the lac operon is called as a catabolite repression. Okay. So where the glucose is actually repressing or is actually suppressing the expression of the lac gene. So is there any question from anyone? Or let's say, see the last part, that is a tryptophan operon. So like lactose is a inducible operon, tryptophan is actually a repressible operon. So here the tryptophan has a negative impact on the expression of the gene. So this is the arrangement of the tryptophan operon. Like there is also operative region, that is the inactive form of the repressor. Okay. So in case of lac operon, what was happening? In absence of lactose, the repressor was remaining bound to the operator region. But here, in case of tryptophan operon, in absence of tryptophan, this repressor is actually inactive. So it is not bound to the operator. So in absence of tryptophan, the mRNA synthesis is continuing. But as the tryptophan will accumulate in the environment, so what will happen? Now the tryptophan will act as a activator for the repressor. So it will go and bind to the repressor. So this will make the repressor active from inactive form. So the active repressor will go and bind to the operator region, because of which the mRNA synthesis will be slowed down. So this is why this can be called as a repressible operon, where the element itself is suppressing the expression of the gene. Now, there is a unique concept of attenuation of the tryptophan operon. So, attenuation is what? Let's understand what is meant by attenuation. So as you can see here, this tryptophan operon, it is having the tryptophan mRNA actually. This is having a leader peptide, or it is called as a leader sequence. LEADER, leader sequence. Okay. As you can see, there are 4 numbers are given here. 1, 2, 3, 4. So these are 4 specific sequences of the leader peptide of tryptophan mRNA. So when the tryptophan is present, so during that time what happens? When the tryptophan is present, the leader peptide is complete, as you can see here. Okay, so here when the tryptophan is present, or when the tryptophan levels are high, the ribosome quickly translate the sequence 1 from the codon reading frame encoding the leader peptide, and it blocks the sequence 2 before the sequence 3 can be transcribed, and the continued transcription leads to attenuation of the terminator, and the attenuated structure is formed by the sequence 3 and 4. So what happens now when the tryptophan is more in the medium, or the tryptophan concentration is high? So tryptophan, when like tryptophan at the high concentration, what it does? Now, it allows, or actually it stops the synthesis of the polypeptide 2. Okay. Now, because of this incomplete synthesis of polypeptide 2, the 3 and 4 cannot be transcribed, and they will form a hairpin of structure like this, a hairpin kind of structure like this. So, because of this, what will happen? The RNA polymerase or the ribosome cannot move, and the gene expression will be slowed down. Okay. As you can observe, the RNA polymerase has been removed from the DNA mRNA sequence because of the presence of this hairpin-like structure by the loop or pairing of these two sequences, sequence 3 and sequence 4. But when the tryptophan levels are low, the ribosome pauses the leader sequence 1, and formation of the paired structure occurs with the leader sequence 2 and 3. But this does not stop, or this actually, because of this, the leader sequence 3 and 4, this kind of hairpin structure cannot be synthesized. So there is no problem for the RNA polymerase to move, and the transcription will continue. So that is how the leader sequence plays an important role in regulation of the tryptophan operon. So attenuation means what? It will try to lower the expression level. So when the tryptophan is present, by forming a hairpin kind of structure between the segment 3 and 4 of the leader polypeptide, the tryptophan tries to block the transcription of the tryptophan operon. But in low concentration of the tryptophan, the leader sequence cannot be completed, and that is why the 2 and 3 will form a loop kind of structure, and the 3 and 4, like this inhibitory 3 and 4 hairpin kind of structure is not formed, and there is no problem for the RNA polymerase to move and transcribe the mRNA. So that is why in this condition, or at low tryptophan level, the gene expression will continue, whereas at the high tryptophan level, the gene concentration will be or the gene expression or the mRNA synthesis will be minimized, which we are calling as attenuation. Okay. So this is one important mechanism to remember, like the lac operon, the catabolite repression is there, and for the tryptophan operon, this attenuation is important. So
Ma'am, in the hairpin loop structure, there will be base pairing between them.
yes, as you can see, there has been complementary base pairing between them. Okay, so this actually as the 3 and 4 will be base paired like this, the RNA polymerase can no longer remain fitted to this. So it has to be removed. So if it will be removed, the further synthesis of this chain cannot take place. So there will be stopping of the transcription process. But here, as you can see, the pair has means the structure has been formed between the 2 and 3, and there has been no complementary pairing, no blocking has been there. Simply a loop has been formed, and because of which the RNA polymerase does not have any problem to move along the mRNA. So it will continue to move and it will continue to transcribe the sequence as well.
Okay, ma'am.
So is there any question from anyone till now, or else we will move to the next topics? Okay, so next topic is all about the sex determination and chromosomal abnormality and all. So let me explain you. So see, simply sex determination we can call to assign a specific gender to a individual, like to assign the means how whether they will be producing the male gamete or female gamete. Accordingly, the sex is assigned either male or female. So there are various ways by which
Ma'am, the regulatory system will always present in every mRNA.
which regulatory system?
A regulatory sequence
No,
will
this
be
is
present in every.
specifically, see, the 3' region of every DNA will have the regulatory part. So, like depending upon which type of regulation is required, the mRNA means some of the mRNA might be having a regulatory sequence where by binding or by modification, the gene expression can be controlled. But this particular attenuation thing that we discussed, it is only specific to tryptophan mRNA. It is not present in others. Okay. So this attenuation thing, it is only restricted to tryptophan mRNA. But the other mRNA will be having other kind of regulatory sequences, like the 3'
Ma'am,
base.
in lac operon, there will be no regulatory sequence in the mRNA.
No, not necessarily it will have the regulatory sequence. It need not have.
Okay, ma'am.
Because every gene has its own way of being regulated. The regulatory mechanism is not same for all the genes. So accordingly, the type of regulatory sequence will vary. So coming to the sex determination, so broadly the sex determination is means it is classified by two ways. Either it will be environmental way or it will be a genotypic way. So environmental means when the an external factor will determine whether the organism will develop into a male or female. For example, in crocodiles, actually, usually what happens if the organism is raised in a low temperature means environment, then mostly they will develop as female, and if they are reared in high temperature region, they will develop as males. So it might be because the genes that regulate in the specific temperature will be determining the gender of the individual. Similarly, there could be genotypic way of sex determination. So, like in case of humans, if we see our chromosomes are responsible for determination of the means maleness and femaleness. So, if an individual is having two sets of X chromosome, it will develop as a female. If it is having X and Y chromosome, then it will develop as a male. Like similarly, there is in Drosophila also we have different type of sex determination mechanisms. In case of honeybee also, there is haplodiploidy concept of sex determination. The haploids they will develop as male and the diploids will develop as female. Here in another example, you can see the C. elegans. If the organism is undergoing cross fertilization, there is a possibility of male and female in a ratio of 1:1, means 50% will develop as female, 50% will develop as male. But if the organism is undergoing self fertilization, there is a ratio of 500:1, means most of them will develop as hermaphrodite, or which is also regarded as a female. So there are various ways by which sex is determined in different group of organisms. So in case of humans, if we say, I already told, the X and Y, the allosomes, they are responsible for the determination of the sex. Particularly, the Y chromosome is associated with the maleness. So it has a specific reason called as the SRY, which is called as the sex determining reason on Y chromosome. That is actually responsible for releasing a factor called as the testis determining factor, which is a transcriptional factor, which is highly essential to determine the sex of the individual. Like during the embryonic development only, the sex is determined. So what is the mechanism? You can have a look how exactly the maleness and the femaleness is determined. So, human sexual differentiation, the undifferentiated gonad means it has not been developed into either testis or ovary now. So, if the individual has a Y chromosome, then it will release the testis determining factor, which will promote the development of testis. So, as the testis will develop, it will have released the testosterone that will develop the male reproductive organs, and it will also have Mullerian inhibitory factor, which will suppress the expression of the Mullerian duct. So Mullerian duct means like that will give rise to future oviduct or female reproductive parts. Similarly, if there is presence of 2X chromosome or there is simply absence of Y chromosome, then it will develop into ovary, and the ovary will it will release MIF, that is the anti-Mullerian hormone, and also testosterone that will lead to regression of the Wolffian duct. Wolffian duct means the male reproductive duct, like your vas deferens, etc. So ultimately, it will lead to the development of the female reproductive parts, and in presence of testis and testosterone, it will develop into male reproductive part. So that is how the male and female gonads are developed in case of humans. In Drosophila, there is an interesting way, that is your genic balance theory is used to predict the sex of the individual, like whether the ratio of the autosome and the sex chromosome is responsible to determine the maleness and the femaleness. So if the X by A ratio is 1, it will develop as a normal female. If it is 0.5, it will develop as a male. If it is more than 1, meta female or super female. If it is less than 1, meta male or super male. If it is 0.5 to 1, it is called as the intersex, means it is neither male or nor female. Okay. So these organisms, they usually do not survive, they die easily. So normal male and female, they usually survive. Then you might ask, what is the role of Y chromosome? So the X by A ratio will determine whether the individual will be male or female. But X by A, let's say, suppose there are 2X chromosome and one autosome. Okay. Sorry, there is one X chromosome and two autosome. So it is 1 by 2, that is 0.5. So it will develop as a male. But since there is no Y chromosome, it will be a sterile male. So in Drosophila, the Y will play the role in fertility of the male chromosome. So the individual without a Y may be a male, but it will not be a fertile male. It will be sterile. So this is the
Ma'am, in Drosophila there will be no fixed chromosome.
no chromosome will be fixed, but the number of autosomes and allosomes may vary. Okay, so means due to that what we can say, kind of polyploidy kind of thing, it may it may become
Ma'am,
very.
as I know, the Drosophila contain 4 pair of chromosome.
Yes. So, means in normal condition, it will be having the same now 4 chromosomes, and the number of X and Y chromosomes will be fitted accordingly. But in different, see, as I told you, during the meiosis, these kind of things might occur. Okay. But as I told you, they will not survive. So that will be determining the sex of the individual, but they will not survive. They will die. So during the during the gametogenesis, due to non-disjunction, the other kind of chromosomal what we can say arrangement can occur, but the organism will not be able to survive. So only whatever normal conditions are there, they will survive only. Okay.
And the male and female due to chromosomal aberration, it can be say.
No, no, it's not aberration. It's their way of determining the sex. See, in normal female, there will be two X chromosomes. But in males, there will be only one X chromosome and one Y chromosome. So here only you can see the difference. The number of X is twice that of the number of the male. Sorry, number of X is twice in female than that of the male. So accordingly, the X by A ratio will be determined. 2 by 4. Sorry, this will be 2 by 2, that is 1. If you are getting the ratio as 1, it will develop as a male. So since we are only considering the X chromosome, in normal condition where you will have a 1 for the female, that will automatically be half for the male. So in normal male will have a 0.5. And whatever more than 1 or less than 1 condition I am telling you, it will be because of the abnormal means non-disjunction of the chromosome during the gametogenesis might occur. That will lead to disturbance, or that will lead to unequal distribution of the chromosome in either of the offspring. So there you will have a variation in the ratio. So in that condition, you can have these abnormal conditions, or you can say the other conditions. But those type of organism with this kind of imbalanced chromosome will never survive. Okay, that is the thing. Okay, so how exactly the maleness and the femaleness is determined in Drosophila? Let's see the genetic mechanism behind it. So there is a gene called as the, sorry, there is a gene called as the SXL gene, which is also called as sex lethal gene. It is called as sex lethal gene. So if the X by autosome ratio becomes 1, in that condition, the SXL gene will be activated and it will activate another gene called as TRA. Then the TRA will activate another TRA2 and they will form two different type of transcription factor, TRA and TRA2, or proteins or transcription factor, whatever you can say. So these two can now regulate the alternative splicing to have a DSX protein which is female specific. So just memorize in the alternative splicing slide I told you, if there is combination of exon 3 and exon 5, this will develop into a female. So this kind of splicing is actually regulated by the TRA2 protein, okay, this transcription factor. Now, if there is the X by A ratio becomes half. Okay. So even though it is having 2X chromosome, but accordingly, if the pair of autosome, if it becomes half, then this SXL gene will not be expressed. So in absence of this SXL gene, no TRA protein will be produced, and this will develop into a male specific protein. That is exon 3 and exon 4 will be joined, and whatever protein will be produced, that will develop the maleness in the fly. Okay. So if this SXL, the ratio here is determining how exactly the gene should be expressed and which type of sex differentiation protein should be produced. So if the ratio is 1, then you will have these genes, this cascade of genes will be activated and female specific protein will be produced. If the ratio is 0.5, then there will be no expression of the SXL gene, and due to absence of this SXL gene, no TRA will be activated, and ultimately the male specific protein will be produced. Okay, so next we have the concept of dosage compensation. So what is exactly dosage compensation is? As you can see that there is a discrimination in the number of sex chromosome that is present in different sexes. Like in case of human also, if you see, there are two X chromosomes in the female, but there is only one X chromosome in the male. So there is a variability or variation in the amount of sex chromosome that is present. So this must be balanced. So how the balancing will occur? Now, the balancing can occur by three different ways. One is X chromosome inactivation or hyperactivation of the X-linked genes in male or hypoactivation of the X-linked genes in female. So, in case of humans, as it occurs, one of the X chromosome is completely inactivated. So, it is now similar to male condition. Male is also having one X chromosome, female is also having one X chromosome. The second X is completely inactivated. Now, the hyperactivation of the X-linked genes in males means since males have only one copy of the X chromosome, whatever genes are present in the male or particularly in the X chromosome of the male, they should be transcribed in twice in rate in comparison to a female. So again, like if there is 100 copy of mRNA in female, the male can produce 200 copies of the male can also produce 100 copies of the same chromosome. Okay. So this is what is called hyperactivation. Means whatever X genes are present, they should be expressed in more amount to balance or to match with the expression of the gene that is present on the X chromosome. Okay. Like if there are two X chromosomes in one of the individuals and together they can express 200 copies of the gene, alone the male X chromosome can produce 200 copies of the gene to match the expression level of the X chromosome with the female. So this is hyper. Hyper means more activated. So similarly, there could be hypoactivation of the X-linked genes in female. Like if the male is having only one copy and the female is having two functional copies of the X chromosome, so both the X chromosome can reduce their gene expression by 50% to match the expression of the X chromosome that is present in single copy in the male. So these are the ways by which the dose can be compensated due to difference number of X chromosome between male and female. So this is the same how the dosage compensation occurs in Drosophila, like the means how the sex is determined. Similarly, the similar genes play a role in the dosage compensation as well. So there is only one specific gene is added, that is activated, called as MSL2, that is male specific lethal. So here what is happening, if the SXL gene is inactive, the MSL2 remains active and it can lead to dosage compensation. Means it will lead to hyperactivation of the gene, X-linked gene. Now if the ratio is 1, so MSL2 is actually it's suppressed or it is degraded, you can say. There is no dosage compensation, means the X chromosome will not be hyperactively expressed. They will remain suppressed. So this MSL gene plays a role in dosage compensation. Now, if we see in case of in case of humans, there is the concept of X chromosome inactivation or the concept of bar body, which is explained by Lyon hypothesis. So LYON. So this could be also an important question for your exam. Lyon hypothesis. So what does Lyon hypothesis exactly tell? The females have two X chromosome, one is paternal and the other is maternal. During early development, there is inactivation of one of the X chromosome in the female's somatic cells. So out of the two X chromosome in females, any X chromosome randomly becomes inactivated except in marsupial and in some extra embryonic tissues of some placental mammals. The X inactivation is a completely random process. And once the X chromosome is inactivated in progenitor cells, the same X chromosome will remain inactivated in all descendant cells. So, what does it exactly mean? So, it means if this is the zygotic stage, let's say the embryo has become multicellular now. So, this is X chromosome. I will be using two colors. Let's say the green one is maternal and the blue one is paternal. So, one of them must be inactivated in order to compensate with the single X chromosome present in the male. Now, which will be inactivated? That is completely random process. So, there is no specific way to tell whether the maternal will remain active or the paternal will remain active. But if in one of the cells, like let's say here, this maternal gene remains active and the paternal gets inactivated. So in all the descendant cell, only the maternal X chromosome will remain active. Similarly, let's say here the maternal is degraded and let's say the paternal remains active. So in all the descendant of this cell, only the paternal X chromosome will remain active. Okay, so that is the meaning of the second point. Though it might be randomly inactivated, but once the active, whatever X chromosome remains active, all the descendant cell will have the same chromosome in the active form or the same X chromosome in the active form.
Then the inactive X chromosome will undergo replication or not?
Uh, yes, it may undergo replication, but it will not be expressed. Okay, so it will not produce any kind of mRNA or anything, but yes, it will undergo replication. There is no problem in that. So as you can see here, this it will remain attached to the cell membrane like this, and this is what is called as a bar body. So inactivation means actually the genes are silenced. So it does not, it will not create any problem to the structure of the chromosome. Simply functionally the chromosome will not be expressed. The genes located on the chromosome will not be expressed. Okay. So this is what is called as a bar body. Okay. So in a normal human female, you will have only one bar body. So next we have the how exactly the mechanism, if we see in case of human beings, how the X chromosome inactivation takes place. So like you can see, there is a choice, means randomly whether either the paternal or the maternal chromosome is chosen. So there is a X inactive specific transcript, like we saw the miRNA. Similarly, there will be an RNA present which will have a complementary sequence to one of the X chromosome. Okay, so since both the X chromosome are homologous and they have exactly the similar sequence, so the 6 can act on either of the X chromosome. It can act on maternal also, it can act on the paternal also. So let's say randomly here the paternal chromosome is chosen to remain active. So the paternal, for the paternal chromosome, the TSIX, that is actually antisense of this cyst, will be present, and there will not be any formation of the silencing mRNA, and the X chromosome will remain active. But here the maternal chromosome has to be silenced. So that is why what will happen? The T's for the maternal sequence will be turned off, so that the X means this X inactive specific transcript or gist can be activated or can be synthesized, which will go and bind to the X chromosome, and the maternal X chromosome will be silenced. So that is how the X chromosome is silenced in case of human beings. So can I continue for 10 or 15 minutes more, or
Yes,
should we
madam.
wind?
Yes, ma'am.
Okay, so yesterday someone was asking, I don't know if he is present today or not, regarding my to share the number and all. So I discussed with the means IGNO official, and they said there is no problem. So you all, if you are interested, you can note down my number and you can add me to the WhatsApp group or whatever you have made. I will share my PPTs there. Okay. If I don't know if someone is in charge of that. Who was asking regarding this to share the PPT over WhatsApp? Is he present? I don't think he's present. So if you all are means added to any kind of WhatsApp group or anything, okay, you can just note down my number. I am writing it in the chat box also. 9938082112. So you can add me in the group. I will share the PPTs and whatever other information.
Ma'am, please repeat the number, phone number.
993808
Yes, ma'am.
82
82
112.
112. Thank you, ma'am.
Okay, so you can add me to your group, and if you need means any kind of communication, we can communicate there also. I will share the PPT and whatever.
Thank you, ma'am.
Okay. So just let's have a few more slides to discuss, and then we can wind up. Okay, let me like proceed for 10 minutes. Please allow me to proceed, or else we will be lagging behind. Okay, so next there comes the cytogenic genetic techniques for studying of the chromosomes. Sorry, studying of the genetic makeup of human beings particularly. So we have the karyotyping. As you can see here, this is the mechanism of karyotyping. You can take out some blood. You can culture the blood, then add colchicine and hypotonic saline solution. Can fix the cell. You can spread the cell, and then you can stain with the Giemsa dye. And since you have applied colchicine, the cells will be arrested at the metaphasic stage. And since the chromosomes are best visible during the metaphase, it is easy to visualize them during the metaphasic stage, particularly for the shape, structure, and number. So, whatever diagram you are getting, that is what is called as the karyotype. Okay. So, karyotype is very important to find out whether there is any kind of structural or functional abnormalities there. Okay, so that way you can identify if there is any chromosomal anomalies there. Similarly, there are various banding techniques are used. So you can go in detail in your material. There is G banding, there is Q banding, C banding, N banding, R banding, T banding. A lot of banding techniques are there. So which will actually help you to locate and to actually identify a particular chromosome. Okay, so here I have cited an example of a G banding and how exactly the banding will help you in studying the chromosome structure. So, as you can observe, when you provide the dye to the chromosome, it can have distinctive banding patterns like this. Okay, so it becomes easier for you to trace whether there has been any kind of structural abnormality in the chromosome or not. So the human chromosome, it is identified into 7 different groups. So starting from chromosome means in a sequential manner, the group A contains chromosome number 1 to 3 that are mostly metacentric. Then group B contains 4 to 5 that are actually submetacentric. I hope you know what is this metacentric and all mean. So metacentric mean when both the arms are exactly equal. Submetacentric, one arm is slightly shorter than the other one. Then acrocentric, one arm is extremely small in comparison to the other one. And telocentric, there is only one arm is present, the other arm is missing. The centromere is at towards the end. Okay. So accordingly, there are different types of means based on the shape, the chromosome has been grouped into various groups. Okay. Then comes other techniques like your okay, so this is what is nomenclature is given. As you can see, there is the arms are given as P and Q. The short arm is given the P and the long arm is given Q. Then within that you have specific banding regions like 1, 2, and 3. And within each banding region you are having sub bands. So whether there has been any kind of insertion or deletion or there has been duplication, that can be easily identified by staining and by karyotyping. Okay. Similarly, there are other techniques like more advanced techniques are there like your spectral karyotyping. This is actually application of a technique called as FISH, fluorescence in situ hybridization. So here, specifically labeled fluorescent label probed or dye can be used, like complementary sequences that are tagged with fluorescence colors or fluorescence tag, we can say. They can be released and they can be allowed to hybridize with specific chromosomes to give each chromosome a specific color, so that it will be easier for you to analyze if there has been any kind of structural or numerical abnormality with the chromosome. Okay, so the probes are actually they are known to you. You know exactly which probe is identical or which probe is complementary to which chromosomal sequence. So accordingly, you give a fluorescence tag to each probe and allow them to hybridize with respective chromosomes. And from the fluorescence means emission that you will be getting, you can find out and you can actually study the morphology of the chromosomes. Okay, similarly, there is also chromosomal microarray analysis, that is CMA. So microarray means at a time you can analyze thousands of samples together in a single run. Okay. So here also you can see this the patient chromosome you have labeled with a green color, and the means normal one you have labeled with a red color. So in whatever you allow them to hybridize, so depending upon which kind of emission you are getting, you can actually predict what could be the possible problem or a normality or abnormality within the chromosomes. So if the color appears like this yellow, that means the normal and abnormal, like the patient is having normal, the suspect is having normal chromosomes, there is no problem. And since the green and red are hybridized, they are giving a yellow color. Now, if you are observing a green color, green means the patient chromosome is obviously more, so means there must have been some duplications. Okay, so that means there is abnormality. So if you are getting a red, means the green is less, so less means there has been some loss of the chromosome, so it might be due to deletion, like that. So from this color pattern, you can actually predict what could be the possible problem, structural abnormality or structural anomaly within the chromosome. Then comes the non-disjunction of the chromosome. So here this is actually, so up to this we discussed the structural abnormality. Here comes the numerical abnormality. Okay, so numerical means actually during when the meiosis takes place, sometimes the chromosome segregation may be improper. That is what is called as non-disjunction of the chromosome. So like if you have heard about the trisomy, monosomy, like the development of Klinefelter syndrome or Down syndrome. So how exactly the chromosome gets like extra chromosome is added, or how exactly the existing means there is a loss of the chromosome. So it occurs due to actually non-disjunction of chromosome during the meiosis. So when there is a change in the single chromosomal number, this is called as aneuploidy. Okay. So as you can see, there are two copies of the let's say this is chromosome number 18, or let's say this is chromosome number 21. So the 21st chromosome is present in 2 copies. So what happened during the first meiotic division? The paternal and the maternal chromosome that did not separate. So both the chromosomes moved to one pole, to one cell, one daughter cell, and the other daughter cell did not have a single copy of the 21st chromosome. Now, when the meiosis 2 occurred, it got normally segregated, the sister chromatids got normally separated. So here you now got 2 copies of 21st chromosome in both the daughter cells, and this daughter cell completely lacked a 21st chromosome. Now, this is a normal gamete having only a single set of 21st chromosome. So, when this gamete will be fertilized with this one, you have 3 copies of the 21st chromosome. This is what is called as trisomy. Okay, so this will lead to Down syndrome. Now, sometimes the non-disjunction can occur at meiosis 2. So, here normally the paternal and the maternal chromosome separated during the meiosis. But during the second meiotic division, what happened? The sister chromatid, they did not separate. So, one gamete received 2 copies of the 21st chromosome, and one gamete did not receive a single copy of the 21st chromosome. So when this gamete is fertilized with a normal gamete with a haploid or one set of 21st chromosome, it also becomes trisomy. Okay. So if the non-disjunction occurs at chromosome meiosis 1, then all the gametes produced are aneuploid. But if the meiosis occurs or non-disjunction occurs at meiosis 2, 50% of the gametes are normal, 50% of the gametes become aneuploid. So a non-disjunction at meiosis 1 is definitely more dangerous than the non-disjunction at meiosis 2. Okay. Anyway, so let's stop here. So whatever portions remained, we will try to finish up tomorrow. And do add me in the group. I will try to share if PPTs and other information, whatever you need, I will try to communicate there. So if there is any other question from anyone, you can ask, or we will wind up the class.
Ma'am, please summarize today's discussion. So please summarize.
Okay, so today actually we started with the mutation. Okay, so mutations, as I told you, there could be 2 types of mutations, spontaneous or induced. So spontaneous means when it will occur naturally due to a mistake of the DNA polymerase. Induced if there are certain bases like your base analog or base modifying agent. So they will actually alter the chemical configuration of the DNA bases, and accordingly it will lead to non-Watson-Crick base pairing. So because of which ultimately the base pair will be different from that of the original or parental base, like means either leading to AT to GC or GC to AT transition mutations, or because of the intercalators there could be insertion and deletions leading to frame shift mutations. Now sometimes the cell, if the error occurs, the cell has a capacity to repair it by either excising a nucleotide or by excision repair, by a repair of the mismatch. If there is a mismatch also, that can be repaired. So there are various enzymatic complex in the cell that will participate in the repair process. Then we had the different type of genes, organization of genes in eukaryote and prokaryote. The prokaryote genes, they are polycystronic, they act under the same promoter. But in eukaryote, they are actually split genes. They are interrupted by exons and introns, where the introns need to be sliced to form the mature mRNA. And it is actually a result of combination of the exons only. Okay. So we had the epigenetic modification. So in order to regulate the expression of the gene, you have to either you can have a DNA methylation or histone acetylation. The methylation actually acts as a suppressor of the gene expression, and acetylation acts as a inducer, or it acts as an activator of the gene expression. Then we have the LAC operon and the tryptophan operon. So both of the operons are actually under the negative control operon, but one is inducible, the other one is repressible. So LAC operon is an inducible operon, where the allolactose, it inactivates the repressor so that the gene expression can take place. In tryptophan, it is opposite. The tryptophan actually activates the repressor to suppress the expression of the gene. In lac operon, the glucose inhibits the expression of the lac gene, leading to a catabolite repression. And in tryptophan operon, there is a attenuation. There is a leader peptide which is present that is actually trying to that actually controls or that actually regulate the expression of the gene. So in presence of tryptophan, the attenuation is more. That means the gene expression is suppressed. In absence of tryptophan, it will be high. Then I guess we move to the sex determination process. Like there are two ways by which primarily the sex is determined, whether it either it is an environmental way where the factors like temperature, salinity, location will determine the gender of the individual, or it could be genetic, like your genotypic combination or chromosomes that will chromosomal constituent that will help in determination of the sex. Then we have different type of techniques, like how exactly you will use the techniques such as your banding pattern or karyotyping, fish or CMA to find out the structural or numerical anomalies in the chromosomal constituent of an organism, particularly human beings. Okay. Then finally, we discussed the non-disjunction process, why this kind of aneuploid condition will occur. So it will occur if there is a non-disjunction of the chromosome or improper segregation of the chromosome or chromatids during the cell division process. So it might lead to aneuploid gamete if the gametes will be aneuploid and they are fertilized with a normal gamete. The aneuploid condition will be sustained and the individual will develop certain kind of abnormality. Depending upon which chromosome is there, it will have like either Down syndrome or Edwards syndrome or Patel syndrome, or in if it is a sex chromosomal aneuploid, it will have either Turner syndrome or it will have a Klinefelter syndrome. Okay. So this is in summary that we discussed, and whatever
Transposable elements are functional gene or non-functional gene?
which one?
Transposable elements.
No, actually, they don't have any specific function in the cell. Okay, but they can influence the cell, means influence the expression of the genes. Because since they are capable of moving within the genome from one place to another place, so they can actually interrupt the certain functional genes. Means they can either silence some of the gene, or they can actually means activate some of the oncogenes like that. So they can be problematic for the cell. But transposable elements themselves do not have any kind of function in the cell. Okay. Means they do not
They
have
are
any.
heritable or not? They
They
are
are?
heritable or not?
Yes, yes. Since they are part of the genome, they will be definitely inherited from one generation to other generation. Okay. So they are part of genome only.
Ma'am, these are placed in a random form in the chromosome.
Yes, actually they will be located somewhere within the, like in some place in the chromosome, but they can change their position from one location to another location. Okay.
They do not have any alternative form.
Alternative form
Means like allelic form or non-allelic form.
no. They will not have anything like that. See, the allele concept is actually applicable to coding part because that is actually giving a phenotype, right? So these are actually non-coding DNAs, means they do not have any particular protein synthesis. Means they will not be transcribed to mRNA or anything. So they are part of non-coding DNA actually, and not part of exon.
Okay, ma'am. Thank you, ma'am.
So is there
Ma'am?
any other query from anyone? Yes.
Ma'am, in tomorrow class, please discuss the question pattern, because in books we have only short question like that filling the blanks and true, false and MCQ. So how questions are coming?
Do you have any access to previous year question paper? I think you are new, means this branch is actually new in IGNOU. I don't know if you have any previous question or not. Actually, I have
Ma'am,
included.
yes, there is a previous year question.
Yes, you can refer to that. What kind of questions will be there? in the if you
50
have
marks. Each paper carries 50 marks. Uh, uh, 5 questions we have to attempt. Each question carrying 10 marks. Two question if there are 2 questions, then 5 plus 5 or
See, here I have an example of your assignment topics. Okay, this was for last year. I don't know if it is same for this year or not. So this is how you will be getting the questions as, and you can see there is a fraction of marks in the right side also. Okay, like which question should be answered for how much marks. So accordingly you can prepare.
When we have to draw the diagram from which mark? I mean, 5 marks, 10 marks, or 2, 3 marks. We have to draw the
See,
diagram.
in biology, diagram is always welcome. Okay, it means in if you compare a text form or a pictorial form, it is always the pictorial form which is more preferred because this gives a clarity. So it depends on you. If you can manage the diagram, then you should. I will suggest you better go for a schematic way of a presentation. Because in text, whatever you write, if you can summarize it in the form of a means schematic diagram, that is much better way of representing your answer. Of
Okay.
course, for 2 marks like that, you need not do the diagrams. But see, if there is something like your flow chart kind of thing or anything like that, like this is forming this and this will lead to formation of this, then that also you can give represent in the form of a flow chart. That will be better. Instead of writing this is forming this, like we today we discussed that Drosophila sex determination thing. So obviously, this looks a way better means of explanation than writing something in text. Okay, so it depends on you how you will be designing your question. So, but in biology, always the diagrammatic form is more means preferred in comparison to the textual form.
Okay, ma'am. Thank you, ma'am.
So, is there any other question from anyone, or we can wind up the class? So tomorrow I will try to wrap up whatever things are left. And
ମ୍ୟାମ୍
yes.
କାଲି ଟିକେ ସିଲେକ୍ଟିଭ ଟାଇପ୍ କ୍ୱେଶ୍ଚନ୍ ଦେଇପାରିବେ?
Selective question means को particular topic रो ना पूरा entire
ହଁ ଏକ୍ଜାମ ଓରିଏଣ୍ଟେଡ୍ ପାଇଁ।
okay ठीक ଅଛି, I will try. Okay.
Thank you, ma'am.
Okay, so anything else from anyone? तो exam actually केबे अछि माने केबे exam schedule?
ମ୍ୟାମ୍ ଡିସେମ୍ବର
ଜୁନ୍ ଜୁଲାଇ ମ୍ୟାମ୍ ନା ଜୁନ୍ ଜୁଲାଇରେ ବି ଅଛି, ଡିସେମ୍ବରରେ ବି ଅଛି, ଦୁଇଟା ଫେଜ୍ରେ ହୁଏ।
ମାନେ ଦୁଇଟା ଫେଜ୍ ମାନେ ସେମ୍ ସେମିଷ୍ଟର ଦୁଇଥର ହୁଏ ନା, ମାନେ ଜଣେ ଦିଟାକ ଦେବ ନା ଜଣେ ଏଇଟା ମିସ୍ କରିଥିବ ଯଦି ନେକ୍ସଟଟା ଦେଇପାରିବ କେମିତି?
ନାଇଁ ମ୍ୟାଡ଼ାମ୍ 1 year କମ୍ପ୍ଲିଟ୍ ହେଲେ ଆମେ ଦେଇପାରିବୁ ଫାଷ୍ଟ ଆଟେମ୍ପ୍ଟଟା, ନେକ୍ସଟଟା 6 month ପରେ ଦେଇପାରିବୁ।
ଓକେ ଓକେ, ସୋ ଏବେ ନେକ୍ସଟ ତମମାନଙ୍କର କେବେ ଆଟେମ୍ପ୍ଟ କରିବାର ଅଛି, ଜୁନରେ ନା ଡିସେମ୍ବର?
ଏବେ ଜୁନ୍।
ମ୍ୟାଡ଼ାମ ଯୋଉ ମୋର ଗୋଟେ କ୍ୱେଶ୍ଚନ୍ ଥିଲା।
ହଁ କୁହନ୍ତୁ।
ମାନେ ଯୋଉ ସିଏସ୍ଆର୍ ନେଟ୍ ଏକ୍ଜାମ ଟା ଆମେ ଫାର୍ଷ୍ଟ ଇୟର କେତେଟା ସେମିଷ୍ଟାର ପରେ ଆମେ ଦେଇପାରିବୁ?
ଦିଟା ସେମିଷ୍ଟର ପରେ ଦେଇପାରିବେ ଫାର୍ଷ୍ଟ ଇୟର।
ଫାର୍ଷ୍ଟ ଇୟର ଦିଟା ସେମିଷ୍ଟାର ଦେଇପାରିବେ।
ହଁ,
କିଛି
ଯେହେତୁ ତାର
ସାର୍ଟିଫିକେଟ
ଭ୍ୟାଲିଡିଟି ନା, ଯେହେତୁ ତାର ଭ୍ୟାଲିଡିଟି 2 to 3 years ରହେ, ତ ମାନେ ପାସ୍ ଆଉଟ୍ ହେଲା ପରେ ବି ସାର୍ଟିଫିକେଟ ଭ୍ୟାଲିଡ ଥିବ, କିଛି ପ୍ରୋବ୍ଲେମ ହବନି।
ଜେଆରଏଫ ର କେତେ ଇୟର ରହୁଛି? 2 year।
ଜେଆରଏଫ ର 2 to 3 year, ଆଗରୁ ତ 3 year ରହୁଥିଲା, ବଟ୍ ଏବେ ମୁଁ ଭାବୁଛି ବୋଧେ କମେଇକି 2 year କରିଦେଇଛି କି ଓଲଟା
ଓଡିଶାରେ
ଏମିତି,
କିଛି
ଆଗରୁ 2 ଥିଲା
ଓଡିଶାରେ
ଏବେ
କିଛି
3
କଲେଜ
କରିଛି। ଆଜ୍ଞା,
ଓଡିଶାରେ କିଛି କଲେଜ ଅଛି କି ମାନେ ଜେଆରଏଫ ର ମାନେ ପିଏଚଡି କରିବାକୁ?
ନା ଜେଆରଏଫ କଲେ ତ ଯୋଉଠି ବି ଆପ୍ଲାଏ କରିବେ ସେଇଠି ପିଏଚଡି କରିପାରିବ ନା, ଫେଲୋସିପ୍ ଟା ତ ମିଳିଯିବ, ମାନେ ସିଏସ୍ଆର୍ କି ୟୁଜିସି ଯୋଉଟା କ୍ୱାଲିଫାଏ କରିବେ ସେଇଠୁ ତ ଫେଲୋସିପ୍ ଟା ମିଳିବ। ତ ଆପ୍ଲାଏ କରିବ ଯୋଉ ବି ଜାଗାରେ ବାହାରିଲା ପିଏଚଡି ପାଇଁ, ତାଙ୍କର ଯେତେବେଳେ ଆଡଭର୍ଟାଇଜମେଣ୍ଟ ବାହାରିବ ଆପ୍ଲାଏ କରିକି ଜଏନ୍ କରିବ, ଯୋଉଠି ବି ହେଲେ ହେବ, କିଛି ଅସୁବିଧା ନାହିଁ। ୟୁନିଭର୍ସିଟି ହେଲେ ବି ହବ, ଇଭେନ୍ ଯୋଉ ଇନଷ୍ଟିଟ୍ୟୁସନ ଗୁଡା ଅଛି ସେଠି ବି ଆପ୍ଲାଏ କରିକି ଯାଇପାରିବ।
ଆଜ୍ଞା, ଥ୍ୟାଙ୍କ ୟୁ।
ठीक अच्छी, any other question or we can wind up the class? Then thank you So tomorrow, let me see how I can wrap up the things. Okay.
Thank you, ma'am.